Welcome to today's class. So, in last class we are discussing about the generation of diversity. We are going to continue again today. So, hope you have already went through the book and more or less you have the idea like what is going on regarding. So, like all other previous classes, I will just spend a minute on the previous slide that I ended up in the last class. Okay. So, the last class we are discussing about the number of variable region of kappa and lambda light chains and heavy chain, diversity region which is not present in the light chain, only present in heavy chain, and the joining segment or joining part is in the light chain and heavy chain. And you know the constant domain is also varying, like kappa chain has 1, lambda chain has 4 to 5 and heavy chain has 9. Though there are 5 different kind of isotypes, there are some subtypes also. We will see most probably today's lecture or maybe the next lecture, this lecture or the next lecture. Okay. Though the constant domain is not doing any taking any part on the receptor diversity, but it is there for their the effector functions of the antibody. Okay. So, this part we have already discussed, like the light chain, heavy chain, and there how many there possible of if V and J. Say for light chain we have, for example, here kappa chain we have 38 maximum and 5 joining chain. So, if randomly they mix up together or join together, there will be possibility 190 different possibilities. So, that kind of calculation what is going to happen and what is possible thing we calculated, and even if I did some mistake in calculation or something, you can calculate. It is a very simple calculation, like for kappa chain what are the variability. If I am taking the maximum number 38, then it is 38 times 5. In case of lambda chain it is 33 times 5, and in case of variable region we have to first recombination is happening between D and J, which is 23 times 6, and with that D J combination B is coming. If I consider 46 is the maximum. So, maximum possibility is 46 into 23 times 6. Okay. So, these are the possible number of heavy chain and light chain. Now, one heavy chain can join either one light kappa chain or one lambda chain. In any antibody, the light chain should be either kappa or lambda. It is not like one is lambda and one is kappa, and once it is done for a particular B cell, that is fixed. So, it is the B cell receptor and antibody is same, that I already told you. So, upon activation the same receptor is synthesized differently and secreted as antibody. So, any B cell receptor, if the receptor, this is the B cell and the pain is the receptor, that receptor will be either kappa or lambda. Okay. So, if we combine like how many possible of total antibody is possible, then we have to multiply this total number of heavy chain times kappa, because any kappa chain can assemble with any heavy chain. Similarly, any lambda chain can assemble with any heavy chain. So, they will combine, that will be the number of kappa containing antibody plus number of lambda containing antibody. This is the diversity. Okay. So, today we are going to see this, how this V and J are going to join, how suddenly the question comes automatically in mind, if there is a recombination, why there are 2 V segment are not joining, why the 2 J are not joining, why all the D's are one after another, why they are not joined. Similarly, why J segment of heavy chain are not joined. So, who regulates or control this thing, or how it is regulated, like one V will join with only one J in light chain and one D will join with only one J chain, and then this D J combination will join with only one V. Why there is no multiple V segment in one antibody hypervariable or variable region, and why there is no multiple D or J. So, how it is regulated, right. So, To understand that, we have to see some detailed nucleotide structure of the VJ region sequence. Okay. Basically, we have to see, or in fact we have to see the flanking region of the V and J and D segment, how it is located. So, if you see carefully, I mean it is known now. So, if you see this is the V region. Okay. So, this is the V region and this is J region. So, I am talking about a lambda chain. Okay. If I am talking about a lambda chain, if you see this is the lambda. So, this is J and this is V. If we go say for say consider just one V segment and one J segment, and whatever we will discuss right now, it will be true for or any V or any J segment. Okay. So, let us see one for lambda chain. So, the V lambda, immediately the flanking sequence in the 3' end, if you see carefully, there is a heptamer. Heptamer means CACAGTG. Okay. This is a fixed sequence, this is heptamer, and after that there is 23 nucleotide. This 23 is the number of nucleotides, 23 nucleotides, which is random, which is non, I mean any nucleotide is possible. After this heptamer and 23, there is another repeat sequence or sequence which is nonamer, that means 9 nucleotides, which is ACAAACC. So, this nonamer sequence, and it is definitely its complementary sequence, you know what should be it is. So, this nonamer and heptamer sequence is very important for this recombination, and this is called recombination signal sequence RSS. Okay. So, this is spaced in case of V, you see heptamer 23 nucleotide, nonamer, again it will continue and then another V segment, then another, but this orientation like heptamer 23 and nonamer, this orientation for lambda light chain is fixed. Okay. Similarly, if you see G chain of the lambda sequence, it is again a heptamer. Okay, it is again a heptamer, instead of 23 here, it is 12 nucleotide. Okay. So, heptamer 12 nucleotide nonamer, and it is in the 3' flanking sequence. This is also the RSS or recombination signal sequence with 12 base pair spacer. So, if I say 12 and 23 is the spacer between heptamer and nonamer, that is going to take a big role in recombination. So, now if you see same way in variable region of kappa chain, we have a heptamer, but there is a 12 base pair spacer, then a nonamer. It is just reverse. In J, it is heptamer, then 23 pair nucleotide spacer, then a nonamer. So, it is just reverse. In lambda chain, it is heptamer 23 nonamer, and in case of kappa chain, the same sequence is with the J segment like heptamer 23 nonamer. It is just reverse. In same way, if we see the heavy chain variable region flanking sequence is very similar to lambda chain, that is heptamer 23 nucleotide nonamer spacer and heptamer 23 and a nonamer. And if you see this D region, which is both side is heptamer, then spacer is 12 nucleotide, then a nonamer. So, what this thing is, I mean this is I do not know how much I am able to confuse you, but go slowly, it is very simple and straightforward. I can assure you will understand very easily. So, if you remember your previous courses like recombination happen with homologous recombination. That means, if two sequences are same, if they can come like this and pair. Okay. So, first recombination what you need to have, I mean what there should be there. In recombination DNA first should be single stranded, and then it will come and make pair with the other or far located some sequence. So, here what happened, these two repeat, suppose this is double stranded, okay, these two repeat become single stranded, and then these will go and compare and make a recombination site. So, there what is normally happen, one protein binds in this sequence, another protein in bind this sequence, they bring them together. So, if this is a long DNA sequence, if you consider this a long thread of DNA, just assume there is a long thread of DNA, here one sequence is say heptamer, here one sequence is heptamer, or here one sequence is nonamer, here another sequence is nonamer. So, if there is a protein which binds to this nonamer and here is another protein binds to this nonamer, and then if these two protein are very good friend and they always wants to stay together, what they are going to do is they will come together. So, what will happen? They will hold this nonamer sequence here, they will hold this nonamer sequence here and bring them together. Okay. So, these two pair, that we will see what is happening. So, then there is automatically a rule, because why 23 and why 12? 23 means, if you remember your basic DNA structure, that is 11.5 actually per turn, 10.5 per turn, sorry. So, this 10.5 nucleotide in one turn, the correct, I mean if you see the old book or old version of any molecular biology or biochemistry book, you may see that there are 10 base per turn, but now the calculation is slightly modified, it is 10.5 bases per turn. So, either 10 or 10.5 does not matter much. What is happening? If there is 12 nucleotide spacer, that means maximum one turn is possible. So, if DNA helix, if you see, that is one turn is complete in 12 nucleotide, but in 23 nucleotide, if this is just almost double, what is going to happen? You can see, if you consider 23 nucleotide, there will be 2 turn of helix in DNA structure, okay? 2 turn of helix in the DNA structure. So, this 23 and 12 is that way important, because which phase of the DNA is seeing by the protein or other part, that is very important. Okay. So, what happened? It was found that there is a rule called 12-23 rule. This is very important to know. Okay. So, the 12-23 rule or 12/23 rule is a gene segment flanked by an RSS with a 12 base pair spacer typically can be joined only to one flanked by 23 base pair spacer. That means, if one heptamer and another heptamer join together, then one heptamer should have a 23 nucleotide spacer, another heptamer should have a 12 nucleotide spacer, then only they can come and join. Okay. I am going back to the previous slide. Here you see this heptamer is spaced by 23 nucleotide spacer. Similarly, if you see the J of the lambda chain, it has heptamer, but 12. Okay. So, this heptamer can join with this heptamer, because one is 23, another is 12. This is called 12-23 rule of recombination, particularly played here. This is discovered. It is sounds very simple, but if anyone is interested how it is discovered or how they figure it out, you can go and find the real research article or paper. It is not that simple, because it is so many experiments to prove like this simple one line statement. So, if this is true, that means no 23-23 base pair spacer containing heptamer will not join. Similarly, heptamer with 12 nucleotide spacer will not join. If, what I mean, if two segment have heptamer both side with 12 and 12 spacer, which you can see in DH. You see the DH part. In DH part, what happened? There is a heptamer spacer, okay, both side, but both are there are heptamer in the both side 5 prime and 3 prime side of the DH segment, and both have 12 nucleotide spacer. So, if this is the case, if there are hundreds of DH, they will never recombine, because 12-12 will not recombine. Similarly, 23-23 will not recombine. So, if you see and go detailed analysis of the sequence, you will see all the V lambda are flanked by heptamer with 23-23 both side. So, 2-23 will not recombine, 2-12 will not recombine. So, only 12 and 23 will recombine, and that is because this turn and other protein that turn part you just draw a sequence and try to understand which side of the draw a sequence with CSC, AG, TG, and then try play with that. So, you study the recombination from previous classes from genetics or molecular biology, okay, or cell biology, recombination mechanism you read, and then you believe my word, like one turn is 10 nucleotide or 10.5 nucleotide, then 23, and write a sequence and then see which turn it is coming facing what. Okay. So, these 12-23 rule, actually this is happening, and that is why the rule made. Okay. So, no 12 will recombine, no 23 will recombine, no 212 will recombine, no 223 spacer containing segment will recombine, and that makes, like, if all DH has 12-12 spacer, I am repeating it several way, because so that you will remember that 12-12 spacer DH. So, how many DH we have? We have see 1 to 23, there are many DH, and all DH which is not given here, if you see, if you zoom it and then you see what is detail, the detail is that every D there is a heptamer 12 nucleotide nonamer, then another 12 nucleotide another 23 nucleotide. So, that is how it will go. So, every DH is flanked by heptamer with 12 nucleotide spacer, and if the 12-23 rule is okay or right, and it is right. So, no 2D will recombine. Similarly, all J in heavy chain is flanked by 23 base pair spacer. So, no J will recombine. So, one is 12, another is 23. So, it is only possible that one D and one J can recombine. Okay. Now, the good thing is, if you see, one D and one J can recombine first. Okay. 1 D and 1 J recombine. So, if this recombination happen, what will happen? So, J and then D, what is there in D? 12. What is there in B? What is the number of spacer? 23. So, again these 23 and these 12 can recombine. Okay. So, these 12-23 recombine, then finally what we will see, with this J will come here, D will come here. So, J and D will be adjacent, then there will be a 12 nucleotide spacer containing heptamer, and VH has 23 nucleotide spacer containing heptamer. These two will recombine and make again 12-23 or satisfy the 12-23 rule. So, this is the reason why this 2D segment or 2J segment and 2V segment are not joined. So, this is normally happen in living system or in biological science. There are every possibility to have some abnormalities or some deviation of this kind of rule, but this is normally happen that these VDJ recombination follow the 12-23 rule. Okay. So, what is happening? So, now I am coming how this thing is happening. So, if you see this is the same sequence what we have seen before. It is what? This is the orientation, say this is one segment V 1 of say lambda. Okay. So, V 1, then V 2, then V 3, V 4, V 5 and then V n, okay, which is around 38, right, in case of lambda. So, V 1, V 2, V 3, and you see this all this heptamer nonamer are having the same color code, but not in that detail of like the previous slide. So, what is happening, this heptamer, the V region is immediate flanked by this heptamer, then a spacer, then nonamer, then spacer, then again another variable region, then heptamer, then spacer, then nonamer, gradually go on. Okay. And there is a J which has the signal of this heptamer nonamer is reverse. Okay. So, it is reverse sequence. Now, two possibility. If I consider that this V 1 and this J will join by recombination, what is happening? One state forward, like V 1 has this direction of the heptamer sequence and J 1 has this direction of the heptamer sequence. You see in this figure, okay, this is all forward. What is happening in this figure? you see this nonomer, two violet sequence come together, okay. And then these two arrow came here. So, this region nonomer region will come, and that I told it is not coming by themselves. Okay. There is one protein or a complex protein or protein complex which hold them. So, what is happening? One is binding in this region in this nonomer, okay. One protein is binding in this nonomer, another protein is binding in this nonomer, they come together. So, whole sequence come together. What is happening as a result? All the internal sequence between or in sequence in between this V and J, they become a very big loop, clear, very big loop where V2 to Vn. That means, if there are 38, one is joining, that means 39 V segment are in this loop with all this sequence. Then what happened? Then there will be a recombination, and recombination you know that there is a DNA cut, then join, right, that I will come later. So, what is happening? They come together, then there is a endonuclease which will cut that DNA, then there will be a joining or some synthesis or repair, ligase, all these things will come. So, what is happening? These two sequence will come together, and this sequence will make a very big loop, okay, and this V and J will join. So, what is happening actually? So, big length of DNA, assume that there is a big thread. So, you make a bring here, then make a tie, given knot here. So, DNA 2 sequence here, bring them together, you make a knot. So, there will be a very big loop. Then if you cut it here, what will happen? These two piece will come, I mean one was here, another was here. So, this will come and the loop part will go out, okay. The same or similar thing is happening. So, here, so the big loop is going and Vj join. One thing we have to remember, mostly or most of the cases or almost all the B cells, and it is happening in T cells also. Once recombination is done, like one Vj joined in light chain and heavy chain Vdj joining is complete. So, the recombination machinery, which is one or several very important proteins which involves in this process or the recombination process, which bring them together, cut it and join it and do that do this job is not expressing. So, recombination machinery in B cell and T cell disappear when heavy chain light chain recombination is happening in B cell, and in case of T lymphocyte alpha and beta chain rearrangement is happening. These machinery or recombination are not expressing anymore, and at the same as a result what is happening, no further recombination is happening. So, once recombination is successfully done and it makes a receptor, either B cell receptor or a T cell receptor, no more recombination is happening. It is normally this is the case. So, one B cell is one done. So, one say suppose in this case what happened, this is very simple and straightforward. What is like V1 is joining with J. So, rest of the V is deleting out anyway. So, there is no scope of further recombination, right, but this is not the always case. It is possible that V3 can join with Vj. So, V3 to Vn will be V4 to Vn will be eliminated, but V1 V2 will still exist in the cell or in the chromosome, but even after that it is not going to recombine anymore. Even if I say the last one, the Vn is recombined with J, okay. So, what will happen? The in between some very little portion of chromosome will be deleted, rest V1 to Vn minus 1, in this case it is say in case of lambda it is 37. So, V1 to Vn minus 1 is still going to be in the chromosome, but even after that no recombination is going to happen. So, once recombination is successfully done, why I am telling successful, that any recombination can lead some non-frame or misframe or deletion addition kind of mutation. So, the protein may not be perfect or some frame shift can happen, then we say that it is not a fruitful recombination. So, fruitful recombination, if it is there, no further recombination is going to happen. Okay. But this is one way that it can happen, it make a big loop, they eliminate. So, they will be completely out from the chromosome forever. But in this case, okay, this is not very it looks little difficult, but it is not that difficult. It is particularly purposefully shown here in this way. If you see this line, what you see is V1, V2, all arrows are in this direction, right. In case of Vn, arrows in this direction. What does it mean? It means that this Vn segment is in the other strand of the DNA. Okay. It is other strand of the DNA, because in chromosome protein or gene may be in one direction, and sometimes you see the arrow is in different direction. It is not that gene will start from 3 prime to 5 prime, because the convention of DNA is what the way we read or we study the gene and everything, we always say 5 prime to 3 prime. So, suddenly one gene is direction this way, suppose this is the chromosome, another gene is direction this way, another is direction this way, but another gene may be this direction. What does this mean? So, that means if this is the double stranded DNA, if this is 5 prime and this is 3 prime, 5 prime, 3 prime, then this means this gene is this direction and this strand, and other gene is this direction, means the gene will, this gene sequence or the sense sequence is this. Okay. It is not that it is reverse, it is same way, other direction. So, all genes are 5 prime to 3 prime. Okay. So, these 5 prime to 3 prime, in this case, in this 5 prime to 3 prime, this particular one, if and this J1 is also in this direction. So, if they want to come in this way, like two opposite facing like this direction. So, we have to make this structure. So, this structure like 2 heptamer is like this direction going outwards, our arrowhead is outwards, then the loop cannot be like straight forward loop. It will be a loop, kind of difficult to think like how it is managed, but ultimately what will happen, they will form this structure, they will join, but in this case what will happen, in this case what will happen, it will not eliminate from the chromosome. Okay. It will not be eliminate from the chromosome, and this whole structure, like whole structure means whole sequence of whatever, suppose this is V n, that means V 1 to V n minus 1, V 1 to V n minus 1, in if it is a lambda, that means 37 V segment will be present in chromosome, but not like previous way. Okay. Not like previous way. So, here also same thing is happening, ultimately what we see L n, V n and J, here also L n, L 1, V 1, J. So, it can happen with any one. So, if the orientation of the gene or the if the V 1 segment is here, so if suddenly one gene is, I mean say for example, I am drawing this again. So, if this is the double stranded DNA 5 prime, 3 prime, 5 prime, 3 prime. So, one gene this, then it may be this. I do not know exactly how this I mean all this orientation I do not remember, like all V how it is located, it may be like this, right. So, some V may be this direction, some V may be this direction. So, whatever the direction, so if it is in the same direction like opposite direction is J, like this form, like this direction is V and J is this, then it will go this way, and if it is other strand it will go this way. Clear? So, this is the two thing, and here one thing I do not know whether I mentioned it, what is this L 1? I never mentioned L 1, okay. L 1, L 2, L n. Okay. So, what this means? L is the leader sequence, L is the leader sequence. I hope you know what is the leader sequence or signal peptide, because any protein goes to, I mean all the proteins are synthesized in cytoplasm, then some protein goes to back to nucleus, some protein goes to Golgi, some protein to mitochondria, some are in the membrane. So, all the receptor proteins are going surface receptor proteins are going to the plasma membrane. So, these targeting, like when protein synthesized in cytoplasm, how I mean how the cell will know where to go, because that signal peptide or leader peptide actually the information is written there. There are some machinery how it will go, but that machinery can understand, say they will see the pool, it is just kind of barcoding. Okay. You know that in if you go to airport, you will see that all the baggage different, I mean say suppose in airport you go to Air India counter, you see in the queue some people are going say from Calcutta. Okay. From Calcutta some people are going to Delhi, some people are going to Bombay, Mumbai, some people are going to Guwahati, Bengaluru. Okay. So, all are in the queue and their baggage is going one after another, but they will all collect it in one place and then someone is there to sort them out. Okay, this bag is for Bangalore, this bag is for Mumbai, this bag is for New Delhi. Okay. So, how they figure it out? Because there is a tag. That tag is the leader peptide. Even after that, all proteins are synthesized in the cytoplasm. So, there is a tag, that is the signal peptide. That signal peptide is recognized, and depending on that, whether it is a mitochondrial signal or it is a nuclear signal or it is a membrane signal or secretory signal, everything is written in this leader sequence, and that leader sequence direct the protein to its place or the proper location. Similarly, all this receptor, whatever the B cell receptor, that is supposed to go to where? That is supposed to go to, if this is the cell. So, all protein synthesized here, they will go to membrane and they will display like this. So, how they will go to membrane? This leader peptide. So, this L is leader, and which is a part of V segment, okay, which is not there, and this is ultimately we will see what is happening, because in antibody also this leader peptide is there, okay, but the leader peptide is located in the N terminal domain of the protein most of the time. Okay. Leader or signal sequence most of the time it is present in the N terminal. It is not 100% cases it is valid or true, because there are some internal signal also, particularly the nuclear localization signal, many times it is present in between the protein or end of the protein, C terminal. Okay. So, these leader peptide are the sequence which is present. So, leader peptide is present for everyone, it is not that it is one. So, every V segment is every V segment have this has this leader peptide before their sequence start. Okay. This will be processed in eventually. So, this is the for this lecture, this is here we will finish, and we will see exactly more detail in next lecture what is happening and how this come together. Okay. So, see you for in the next lecture. Bye for today.